Before we start, I'd like to acknowledge that we're meeting here on land that's traditionally owned by the Wurundjeri people of the Kulin Nation, and I'd like to pay my respects to their elders, past, present, and emerging, and also welcome all other people from Torres Strait Islander and Aboriginal communities who work on this campus and are here with us today. So it's my great pleasure to introduce Valentin today. Valentin contacted me a bit over 4 years ago to do a PhD in my lab. Now, my lab is not really a thing, as you know, so it was really a co-supervision with John and I. Co-supervision means John paid, and I supervised in many instances. No, I think it works really well. So, Valentin did his undergraduate in the University of Constance. Constance is a small town just at the border of Switzerland, so that was great to have some Swissness in the lab. He did his undergraduate in the lab of Tommy Brunner. Some of them might know him. He was my own PhD supervisor, which is sort of nice, closing circle. Tommy also has the same hairstyle as I do, so the prospects of Valentin are not great. I think he has much better prospects when it comes to his future, his professional future. When he started in the lab, we really embarked on a new type of project. We wanted to look at intracellular signaling complexes in a way which hasn't been done, at least not here at the Institute, and we were really sure that's going to be a great, easy project which we will pump out great results. But of course, it isn't always as easy. The last years have been hard work for Valentin, optimizing lots of protocols, and I'm sure he's gonna tell you about all this. So there were lots of hurdles we had to overcome and to really answer the question we wanted to tackle. But I have to say, really, in the last year, a beautiful story came together, also because Valentin really worked very hard and put the extra effort in the last years. So that has been really nice. So I'm really looking forward to your talk now. And yeah, please welcome Valentin. Thank you.
Good afternoon, everyone, and thanks for coming to my completion seminar. And I would like to give you an introduction into the pathways of the NOD-like receptors, NOD1 and NOD2, and describe to you some of the molecular concepts that we were able to discover using FLAG-RIPK2 and HA-NOD2 knock-in mice. And in the first part, I will really just quickly touch introduction into inflammation and to NOD-like receptors in general. I will then show you my characterization of the knock-in mice. And the last part really will deal with RIPK2 interactions and most of all RIPK2 post-translational modifications. And during these experiments, I believed we discovered a new regulatory interface on RIPK2. So to highlight the importance of inflammation today, I'm showing you a picture that I took from the Global Burden of Disease study, which is a massive ongoing study led by the WHO. And one of their conclusions was that more than 50% of all early deaths in the world today can be attributed to diseases that have a chronic inflammatory component, like ischemic heart disease or stroke, or also COPD. And the projections for the future really are that the prevalence and the incidence of these diseases are further on the rise. Now, what this also suggests is that there already must be a huge market for anti-inflammatory medications. And again, I think this confirms this very nicely. So if you look at the top list of drugs by market sales on the United States market in 2018, you see that on top of this list really are quite a few drugs that specifically inhibit immune signaling pathways. And on top of this list, really anti-TNF antibodies, and these are used for the treatment of inflammatory bowel disease or also arthritis and psoriasis. Now, another class of immune signaling receptors or pathways that also offer great therapeutic potential are the pathogen recognition receptors. And they're grouped here into two groups. First, the membrane-bound PRRs. This group is mainly composed of the TLRs. And the TLRs, they can recognize different pathogen-associated molecular patterns, PAMPs, but also danger-associated molecular patterns, DAMPs, such as ATP or cell-intrinsic proteins like heat shock proteins, and also DNA. And on the other sides really are the cytosolic PRs, and this list mainly consists of the NOD-like receptors, which mostly detect PAMPs. Now what these NOD-like receptors all have in common is this central NACHT or NOD domain, which stands for nucleotide binding and oligomerization domain. And this domain really mediates conformational changes of the proteins. And they are typically grouped into different subgroups via their interaction domains. So on the right side, you have the pyrene domain containing NLRs, which is a very famous group that can form large inflammasomes. And what we are specifically interested in are the card-domaining NLRs, NOD1 and NOD2, and they are sensors of bacterial peptidoglycan fragments called BAP or MDP. Now, due to their card domains, these proteins were initially thought to be involved in cell death pathways. And early studies also showed that overexpression in different cell lines could induce cell death. However, what we really know today is that they mediate pro-inflammatory antibacterial signaling. And this is a current model, how we understand the signaling pathway. So in an inactive state, NOD2 resides in the cytosol in a close conformation, which is stabilized by ADP. And upon binding of ATP and a ligand like MDP, this conformation opens up, and NOD2 is able to oligomerize directly in the cytosol or at bacterial entry sites, such as early endosomes. It then recruits this very vital downstream adapter RIP kinase 2 or RIPK2, and together they're believed to form larger oligomers. And now what really drives the inflammatory downstream response is the generation of massive polyubiquitin chains, and amongst them are mostly K63 and M1-linked chains, and different E3 ligases have been shown to be involved with this, and amongst them are XIAP and the Lubeck complex. Now this leads to the recruitment of downstream kinases and subsequently to the activation of transcription factors such as AP1 or NF-kappa-B, which then induce a pro-inflammatory response, which also includes the secretion of cytokines such as TNF-alpha, IL-6, or also MCP1. Now, like many other immune signaling pathways, the NOD-like receptor pathways are associated with a diverse range of inflammatory diseases, and most notably really chronic inflammation of the gut, like inflammatory bowel disease. And NOD2 is the top genetic cause to develop Crohn's disease. But what recent studies really suggested is that the pathway is also associated to type 2 diabetes, as well as allergic asthma. And we were able to generate a nice bit of data with the group of Reese Allen, especially with Christine and Nadia, but unfortunately, to the lack of time, I can't show you this today. Now the pharmaceutical industry really has acknowledged the potential of this pathway, and they have tried to develop specific inhibitors, and their primary target always has been RIP kinase 2. And the main reason for this, or the main reasons for this, really are that RIPK2 is absolutely essential for signaling through NOD1 and NOD2. And I'm showing here an in vivo experiment that we did. So we injected mice IP with MDP, and then measured secretion of cytokines into the serum, and you can see that wild-type mice are able to or secrete TNF and IL-6 in the serum, while RIPK2 knockout mice don't respond to MDP challenge at all. RIPK2 is, being a kinase, it's obviously also targetable with kinase inhibitors. It is thought to be a relatively safe target. First of all, we don't know of any other signaling pathways where RIPK2 is involved in. And what RIPK2 knockout mice suggest is that a complete deficiency of RIPK2 doesn't lead to a phenotype under normal housing conditions. Yet there are really no specific drugs available for the use in clinics. And I think this is also highlighted by this very recent phase 1 clinical trial led by GSK for the test of a novel RIPK2 inhibitor in inflammatory bowel disease. And they had to terminate this study due to non-clinical toxicology findings. And the reason why we don't have any specific inhibitors yet is mostly because very important mechanisms of the pathway have only very recently been described. And one major reason for this is that the pathway is quite tricky to study. So first of all, we don't have many reliable antibodies. First of all, we don't have any antibody for NOD2 still. So what I'm showing here is the best NOD2 antibody that we have available, which is published a couple of times, also in very high impact publications. And you can see here lysates of wild type and NOD2 knockout BMDMs. And we detect a band at the expected size, but you can clearly see that this is unspecific. So scientists have been forced to work more with overexpression studies. And in this experiment that I did here with THP1s, I think you can nicely see that this is also quite tricky because it leads to a lot of artifacts. So first of all, wild-type THP1 cells respond very nicely to the synthetic NOD2 agonists L18-MDP by inducing NF-kappa B and MAP kinase pathways. But what you can also see is that after doxycycline-induced overexpression of NOD2, this is really sufficient to trigger pathway activation. And what is very strange is that the cells are subsequently insensitive to MDP treatment. Now this is really where my PhD wanted to pick up, and we wanted to establish novel methods to investigate the NOD signaling pathways at endogenous levels, and therefore we generated knock-in mice. And we wanted to use these mice to determine interactions with inactive NOD signaling complexes, as well as really look at post-translational modifications of RIPK2 because they are a critical regulatory element of the pathway. I don't want to bore you with too many details here. I just want to point out that the generation of CRISPR knock-in mice really has become a standard routine, and all you need is recombinant Cas9 together with a very good guide RNA and a short double-strand oligonucleotide or a plasmid that also encodes for your sequence of interest as well as for homology regions. And using this, we introduced a single flag onto RIPK2 or a single HA tag onto NOD2. And this all is just injected into fertilized single-stage embryos, and after breeding out the mice, you often end up with perfect knock-in alleles, which you can see here in blue, but also very often with different kinds of indels leading to functional knockout mice, seen here in orange. So essentially you will get 2 mice for the price of 1. And As a first step with these mice, mostly because, or also because they were really the first affinity tag knock-in mice that our lab worked with, I wanted to be very rigorous and test whether the proteins were still functional and if they were properly expressed. So to look for their expression, I first did Western blots, and you can see on the left side HA-NOD2 knock-in mice, and I took organs from these mice as well as wild types, which I used to control, and I probed lysates from these organs with an HA antibody, and you can see for HA-NOD2 and also for FLAG-RIPK2 that we are not able to detect the protein with these antibodies. However, after performing immunoprecipitation, you can very nicely see an enrichment of these proteins, and you can see that the antibodies now act specific. And what you can really take away from this is that NOD2 and RIPK2 are widely co-expressed, as you can see here, and that they are most abundant in organs that naturally harbor large immune cell populations, like the intestinal tract, and also the lung or the spleen. As a next step, I really wanted to characterize the expression in more detail and ask which cells specifically expressed the proteins. And for this, I did immunohistochemistry and immunofluorescence. However, detection of the protein was again quite tricky. But I did get a specific staining of RIPK2 using a FLAG antibody in the lung. And you can see sections of a knock-in mouse on the top and the corresponding wild-type controls on the bottom. And it really looks like RIPK2 is expressed in the endothelium of the lung. Now looking at this a bit more in detail with immune fluorescence, you can see that in addition to the endothelium, RIPK2 is also present in the epithelium, while in the wild-type controls, this FLAG staining that I used was really reduced. Now to test the functionality of the proteins, I used bone marrow-derived macrophages, and these cells need to be primed with interferon gamma. We're not 100% sure what it does, but we hypothesized that it induces the expression of NOD2 as well as leads to an effective uptake of NOD2 ligands. And then upon stimulation with MDP, I first measured activation of NF-kappa B and the MAP kinase pathways. And what we're really looking at is degradation of this inhibitory NF-kappa B subunit IKB alpha, as well as its phosphorylation, and phosphorylation of the NF-kappa B subunit P65, as well as phosphorylation of different MAP kinases. And first, I'm showing you Westerns from FLAG-RIPK2 BMDMs. Again, you can see we cannot detect the protein with a FLAG antibody, but what you can see quite nicely with a RIPK2 antibody is actually that the wild type as well as the FLAG knock-in allele are expressed at equal levels. And what was also very satisfying to see is that like the wild type BMDMs, the knock-ins responded to MDP by inducing NF-kappa B and MAP kinase pathways, and this really happened at equal levels. And on the right side, Westerns from the HA-NOD2 knock-in mouse, again, they responded normally to MDP treatment by inducing NF-kappa B and MAP kinases. And what was additionally very nice to see is that for the first time, we were able to detect NOD2 using a HA antibodies in a simple cell lysate. Now, what really happens downstream of NF-kappa B and MAP kinases is the production of cytokines. And I quantified these first in BMDMs, and you can see here in magenta wild-type BMDMs as well as in green and purple, the knock-in BMDMs, that BMDMs produced equal amounts of cytokines upon stimulation with MDP. And to be even more rigorous in testing the functionality of the proteins, we conducted in vivo experiments. So we injected mice IP with MDP, and again measured cytokines in the serum, and again the same pattern as in the experiments before can be seen here as well. Wild type and knock-in mice really respond equally to MDP treatment. Now the conclusions we took away from this were that the addition of these single affinity tags on RIPK2 and NOD2 did not in any way affect protein function, and they responded normally to NOD2 activation, as well as the detection of these single affinity tags in some assays was quite tricky. Now I really wanted to go ahead and do some more exciting experiments, and a large part of my PhD actually dealt with identifying NOD2 and RIPK2 interactions, and to really test whether our system was sensitive enough to detect interactions. I had a look at this interaction between RIPK2, NOD2 and XIAP, and this has been shown in overexpression studies, but really hasn't been confirmed in endogenous experiments. And to test for the interaction, I utilized BMDMs from the now intercrossed mice, so they express FLAG-RIPK2 and HA-NOD2 at the same time. And you can see here in FLAG pulldowns from these BMDMs that even in an unstimulated state, RIPK2 and NOD2 interact. And what you should be able to see is also that XIAP is also present here and co-precipitates together with RIPK2. And that after stimulation with MDP, this really enhances the interaction. And yeah, from this we really concluded that during a steady state there must be some sort of weak interaction between these proteins, and that during signaling these proteins interact more strongly, and RIPK2 as well as NOD2 get ubiquitinated. So for RIPK2 this has been shown numerous times, and this small smear here, this suggested that it was also happening for NOD2. And I was actually able to confirm this in subsequent experiments, but this is not going to be part of this talk. Now, I really wanted to go ahead and I wanted to look at novel interactions using mass spectrometry, and I spent a tremendous amount of time optimizing methods and protocols. But I think in the end, we really had to come to the realization that these single affinity tags are probably not specific and sensitive enough for these sort of mass spectrometry experiments. And this very, very overloaded volcano plot here, I think, illustrates this quite well, because what it shows is the specific as well as non-specific RIPK2 interactions in MDP-stimulated BMDMs after doing FLAG pulldowns. And you see that RIPK2 is enriched, and you have also many other proteins enriched. However, everything that is highlighted here in dark green really are non-specific interactions with FLAG antibodies. And what you can also see, or I should probably say what you can't see, is NOD2 and XIAP on this list, which I just confirmed should be there by Western blot. Now, if we filtered out all these non-specific interactions, what we were really left with was this. So RIPK2 and only a single significantly enriched protein, which very unfortunately also turned out to be another false positive. So to sum this very short section up, I was able to show that NOD2, RIPK2 and XIAP interact at endogenous levels in a stimulation-dependent manner. And I think this is quite nice, and no one else has reported this so far. However, the detection of interactions by mass spectrometry really was not possible. And one reason for this, I think, is that endogenous level, these single affinity tags are just not specific enough. Now, for the last part, I want to describe to you something that worked very well with the mice. And this was determining post-translational modifications on RIPK2. And I found this actually very interesting because these PTMs on RIPK2 are what really regulate this inflammatory response downstream of the NOD complex. And this is kind of how we imagine an active NOD signaling complex. So you have NOD2 and RIPK2 is heavily ubiquitinated, and many different ubiquitin-E3 ligases have been suggested to be part of this, and also different deubiquitinases. However, only the role of XIAP and LUBAC has been confirmed in vivo. And what they do is they generate K63 and M1-linked chains on RIPK2. Now, much less is known about the actual sites that are modified on RIPK2. And the only site that repeatedly comes up in literature is this Lysine 209. And this was originally identified by Hasegawa and colleagues. And what he did is he systematically mutated all conserved lysines in the RIPK2 kinase domain. And he found that mutants of this Lysine 209 didn't signal and also were not ubiquitinated when overexpressed in tuna refugees together with ubiquitin. However, what he didn't find, and what is still not available, is actual proof that this site, in fact, is ubiquitinated, for instance, by mass spectrometry. Now, to look at this from a very unbiased perspective, I was able to isolate modified RIPK2 species using this two-step enrichment protocol that I'm illustrating here. And this works by, you have a protein lysate. So in this case, I used MDP-stimulated BNDMs. And within this total lysate, you have many non-ubiquitinated proteins, but also ubiquitinated proteins. Amongst them is RIPK2. You can enrich for these ubiquitinated proteins using ubiquitin binding domains, such as UBA1. And you can also natively elute this again from the beads that you use for it, and then subsequently do a FLAG pull-down. And what, in theory, you should end up with is a very clean pool of ubiquitinated RIPK2. And the Western blot on the left side really confirms that this worked very well. So I just want you to focus on this ubiquitin panel here. You can see that in the input, there's a faint smear detectable, which is characteristic for ubiquitin Western blot of a whole cell lysate. And after UBA pull-downs, this is really enhanced. And after the subsequent FLAG pull-down, this smear is reduced into a more distinct smear, which almost looks identical with what we detected with a RIPK2 antibody. And this already really suggested that this final sample that we submitted for mass spectrometry consisted predominantly of ubiquitinated RIPK2 species. And this was actually the case, and we were able to determine different ubiquitination sites on RIPK2 across the whole protein, and most of them were dependent on MDP stimulation. And we also found different phosphosites. However, not that many of them were actually dependent on MDP stimulation. And using the data set of a friend and colleague who's now in Munich, Chase Stafford, we were actually able to confirm that these modifications, so at least the ubiquitination modifications happened also in human cells. So what he did is a dicla enrichment for us, and he found essentially the same sites or the corresponding human sites in THP1s. So I went ahead and I generated all sorts of mutants. And my main criteria for this were that the mutations had to be stimulation-dependent and also relatively well conserved. And in addition to all the sites that I detected, I also made a mutant of this putatively super important lysine 209. And to test whether these mutants were functional, I expressed them in RIPK2 CRISPR knockout THP1s using a doxycycline inducible system. And I first had a look at their ability to produce IL-8. And you can see that here wild-type THP1s up on stimulation with L18MDP produce a bit of IL-8. And what you can also see is that all mutants that I tested also produced IL-8 upon stimulation, with the exception of this lysine 209 to arginine mutant that I made. Now, next I looked at NF-κB activation of these mutants. And again, in this massive Western, what you can essentially see is that, like wild-type RIPK2, all mutants respond normally to MDP treatment by inducing MAP kinases and the NF-κB pathway, except lysine 209. And what I also tested is RIPK2 ubiquitination, and a reason for this was obviously that many of the mutants I generated were mutations of ubiquitinated sites. So I really wanted to ask whether the removal of these single sites had an effect on total RIPK2 ubiquitination. And for this I did UBA and tube pull-downs again. And what you can see again is that single site mutation doesn't really have an effect besides this lysine 209. However, lysine 209 still has ubiquitin chains, right? This is only a reduction in ubiquitin, some sort of condensed ubiquitination. Now, since this really was the only mutation that had an impact on RIPK2 ubiquitination, I wanted to ask the question, what type of chains were still present here? And I put an emphasis, of course, on K63 and M1-linked chains, since they are really what drive the pro-inflammatory response downstream of RIPK2. And for this, I used these K63 and M1-linked specific antibodies, and again, used them in pull-down experiments. And what you can see is that wild-type RIPK2 has both K63 and M1-linked chains. However, these chains are really still present with the K209R mutant. So since these chains were still present, this really didn't explain why the mutant didn't signal at all. So to sum this up, I think we found that single phosphorylation and ubiquitination sites on RIPK2 are not required for signaling, as in you can mutate single sites, and most likely they can be substituted for by other modifications. However, the mutation of this putatively ubiquitinated lysine 209 really makes an exception. And this is characterized by cells that express K209 mutants, don't respond to NOD2 stimulation, and also have reduced ubiquitination of RIPK2. However, what was very strange, both K63 and M1-linked chains were still present. So what really made this site special? And to ask this question, I first had a look at the RIPK2 crystal structure, which is seen here, and the RIPK2 kinase domain in complex with ponatinib. And you can see that lysine 209 is at the C-lobe of the kinase domain at this intersection between the alpha E and the flexible alpha E F helix. And if you look at the electrostatic interaction potential there, it really looks like lysine 209 is part of what looks like a hydrophobic pocket, which we believed could be an interaction interface. And looking at the sequence conservation around lysine 209, you really see that pretty much all amino acids there are highly conserved or only substituted for by other very similar amino acids. So from this, we took the hypothesis into account that it is actually this whole pocket rather than the lysine 2,9 alone that is important for RIPK2 function and not signaling. And to test this, I generated more mutants around this pocket, and what you see here in this Western blot, again, is NF-kappa B activation, and I did a conservative mutant of this isoleucine 208 to avaline, and this, so first of all, the lysine 2,9R, as shown before, doesn't signal. However, this conservative mutation of the isoleucine 208 here to avaline didn't really affect RIPK2 function. But what was very interesting and exciting is that mutations of this isoleucine 212, which is directly deep in the pocket, seem to have an impact. So first of all, conservative alanine and methionine mutations responded normally. However, a non-conservative mutation to aspartic acid didn't respond to MDP stimulation at all. And I could also confirm this here looking at cytokines. Again, lysine 209 doesn't produce cytokines after stimulation, and the conservative mutant I28V does, while this non-conservative mutation of this isoleucine 212 to an aspartic acid doesn't produce cytokines at all. And what was additionally very interesting to see is that this other conservative mutant, isoleucine 212 to alanine, consistently produced more cytokines than wild-type RIPK2. So it could be some sort of hyperactive mutant. And next I looked at RIPK2 ubiquitination. And you see on the top panel here in the tube pull down that unstimulated cells, RIPK2 is not ubiquitinated. And then upon treatment with MDP, you get ubiquitination of RIPK2. And this is really reduced with the K29R mutant and is actually totally gone with the I212D mutant. Now, this was a very exciting result for us, and the conclusions that we took from this is that, first of all, lysine 209 was part of a hydrophobic pocket in the C-lobe of the RIPK2 kinase domain, and that this pocket exhibits high electrostatic interaction potential, and also that mutations of amino acids within this pocket could abolish RIPK2 activity as well as ubiquitination. Now, the really important questions were, what is the mechanism behind this, and is this pocket actually an interaction interface? So first, I wanted to be very rigorous and test whether these mutants were actually still stable, because that would be a very easy explanation why they don't signal at all. And for this, I utilized this thermal shift assay that is usually done with protein inhibitors, and what it essentially assesses is protein solubility with increasing temperature. So I took cells in solution, incubated them at increasing temperatures, and then the theory is that more soluble proteins will denature more slowly. And if you then centrifuge these cells and look what's left in the supernatant, you could really assess the solubility and the stability of the proteins. And this massive Western here again shows that with increasing temperatures, wild-type RIPK2 was less stable. And it also shows that all tested mutants were equally stable, besides this isoleucine 212D, which was slightly less stable, but expressed normally and was also as stable at 37 degrees. So from this, we concluded that reduced stability most likely is not the single cause for our phenotype. And we really wanted to ask the question whether this pocket was an interaction interface that is required for NOD signaling. And one idea was that it could be an oligomerization interface. So oligomerization of RIPK2 has been shown to be, or is suggested to be, important for signaling, but in the context of a cell, this was never really confirmed. But then obviously it could also be a binding site for another important protein, and our best bets here were E3 ligases like XIAP and LUBAC. So to test whether this was an oligomerization interface, I generated recombinant full-length wild-type and K2-9R RIPK2 in 293Ts, affinity purified that, and ran it on size exclusion chromatography and compared the elution profile and ran all of the fractions on Western blot. And what you can really see here is that wild-type RIPK2 is present in fractions with lower numbers than K2-9R RIPK2. And what it suggests is that wild-type RIPK2 is able to form larger complexes than the K2-9R mutant. Yet we really didn't want to exclude that this pocket was a binding interface with something else. And our best bet here was XIAP. So XIAP is known to bind to the RIPK2 kinase domain via its BIR2 domain. And there's a binding interface already suggested for this, which involves this aspartic acid residue here. But it's easily thinkable that XIAP binds to RIPK2 at multiple sites. And to test whether our mutants still bound to XIAP, I expressed this BIR2 domain of XIAP, bounded to beads, and incubated these beads with lysates of Th1s that expressed wild-type RIPK2 as well as the mutants. And what you can see quite strikingly is that, yes, indeed, our positive control, this aspartic acid 41 mutation doesn't bind. However, both of the mutants that didn't signal in my assays, the lysine 2,9 and the isoleucine to aspartic acid also didn't bind at all. Now, what I hope, what my data really suggests is, I think, an update to a very old model. And what this model essentially says is that upon NOD2 stimulation, you get ubiquitination of RIPK2 on this crucial lysine 2,9 residue. And this ubiquitination is sufficient to activate NF-kappa B and to produce cytokines. However, what my data really suggests is that there is a hydrophobic pocket in the RIPK2 kinase domain, and lysine 2,9 as well as isoleucine 212 seem to be key residues there. And upon stimulation of NOD2, you get binding of XIAP to RIPK2, maybe directly to this site. And this could be mediated by RIPK2 oligomerization. And subsequently, you get ubiquitination of RIPK2 on multiple sites, maybe including this lysine 2,9. And this really triggers NF-kappa B activation and cytokine production. To conclude my findings, I want to show, I think I've showed you that affinity tag knock-in mice are a great model to investigate signaling pathways. However, that for the detection of lowly abundant proteins at endogenous levels, more specific tags should be used, or triple tags. And our lab is now using triple flag tags, which turn out to be much better than the single flag. On the other side, we failed to identify RIPK2 and NOD2 interactions by mass spectrometry. And in the time of doing all these experiments, I, however, was able to establish different kinds of protocols that can now be used with these or with different affinity tag knock-in mice. I was able to map phosphor and diglycides on RIPK2, so phosphorylation and ubiquitination sites on RIPK2, and I characterized their impact on NOD signaling. And I believe we identified a novel regulatory interface on RIPK2 that is important for RIPK2 function by mediating binding with the E3 ligase XIAP as well as RIPK2 oligomerization. And with this, I'm already at the acknowledgements. First of all, I want to thank all my supervisors, Uli, John, and Andrew, who are really great supervisors and are really scientists someone can only look up to. And I want to thank my committee members, Melissa, Jacqueline, and Richard, who were always very helpful in our conversations and provided great feedback. I, of course, want to thank the CRISPR lab and foremost Marco and Andrew, who of course, generated these mice. A very special thanks goes to Laura, who did a tremendous amount of work for me and ran all of my hundreds of mass spec samples. And of course, I want to thank everyone from Silk Lab. I want to thank Jay Stafford, who has been a good friend and also a good collaborator. I want to thank my lab buddies, Jono, Daniel Frank, as well as Lynn. I want to thank the Reese Allen lab, and particularly Christine, as well as Nadia, who did the asthma challenges, the asthma experiments for me, which unfortunately I couldn't show today. I want to thank Finn Hansen from the MEN lab in Munich, who did the dicli enrichment. And of course, I want to thank the animal techs. Without them, none of this would have been possible at all. I also want to thank Philippe, and I want to thank my amazing girlfriend, Eliza, who always provided support for me in very difficult times.
All right, questions? I have this thing here, but maybe we don't need it. Yes, Robert.
Just a technical one then. So with your mass spec, as I understand it, your control would have been just your untagged control mice with the FLAG antibodies. Why didn't that fish out the non-specific binders to the FLAG antibody?
Well, it essentially did. I mean, it definitely fished out most of the non-specific interactions.
So in your volcano plot where everything was skewed towards your test?
Yeah, so that volcano plot that I showed, what I'm comparing the BNDMs from the FLAG knock-in mice that were either spiked with triple FLAG peptide or left unspiked. And using this spiking with the FLAG peptide, we were really able to determine the subset of non-specific interactions with the FLAG antibodies.
So in both of the final models, the old one, the one that you updated, there was an arrow at the bottom that led to an NF-kappa beta activation. So can you say anything, do any of your experiments give you any hint about what that mechanism might actually be?
I mean, there's evidence that generation of these hetero K63 and M1-linked chains leads to the recruitment of master activators of the NF-kappa B and AP1 transcription factors, like the IKK complex. But I didn't really address that. Yeah.
Good.
For your last aim with the pocket that you identified, you first said that your E3 ligases that might bind there are XIAP and LUBAC, and then you only looked at XIAP later. Did you do any experiments on LUBAC to see if that binds over there?
I did not know. Those were very last minute experiments, and we didn't address any other E3 ligases. Yeah.
So I thought it was quite well established how Beer domains interact with other proteins, right? So do you actually have that sequence, or is that does it resemble a Beer interaction? What you predict there?
I don't think it resembles the typical sequence that you would expect. Yeah.
So the other thing is that when you mutate a residue to an aspartate, and that is right next to the lysine residue, that you know has a really, really strong effect. Don't you expect that there's some kind of effect on the lysine? So this is going to definitely change the pK of the lysine, for example. So is there any way that you can think of how to address that?
Yeah, that's a good question. I mean, I think one of the next steps in this whole story would really be to look at the structures of the different mutants, but so far I cannot comment on that.
So when you analysed your novel mice, you showed that the proteins were expressed by epithelial cells and endothelial cells, is that right? In the lung?
Yeah, we think so.
And then in your mass spec, you did bone marrow maps?
I also did lung.
You also did, okay, stromal cells, okay.
Yeah. We didn't find anything in the lung either. Yeah.
Okay. Have you ever done chimeras to show that the expression of these proteins, the responsiveness to MDP is dependent upon the parenchyma versus the bone marrow compartment?
You mean specifically in the lung now? For
Oh no, just generally.
No. No, I haven't done this, but there's evidence that also other cells, endothelium and epithelial cells, also get activated through NOD2 ligands, produce cytokines. So it's not. I think it's not only the hematopoietic compartment that mediates these responses. Yeah.
In the beginning, when you tagged your mice with HA and FLAG tag, you showed different tissues where the proteins are expressed, and most of them were either both were expressed or none of them were expressed, but in the brain, I think only one of them were expressed.
Yeah.
And you mentioned that RIPK2 is only relevant for node signaling. So do you have an idea why only RIPK would be expressed in the brain and not?
Yeah, I think it was actually only NOD2 in the brain, but then it was only RIPK2 in the kidney. And yeah, I mean, I talked to Uli about this. I have no idea. Maybe it's an organ where we could look at to investigate alternative roles of RIPK2. Yeah, but I don't know it, no.
All right, then John, last one.
Yeah, we got time. Also, it's kind of flipping back to David's question at the back. So you had your 2008 model where the lysine is ubiquitinated, and then you've got your model where it isn't, but then you say, but maybe it is. Now, why are you sort of hedging your bets? David's point is that if you have a aspartate, so a lysine that could interfere with the PK of that residue or whatever. But the fact is you made the lysine to arginine mutation, and that didn't really affect ubiquitination. So you would have to say that the lysine to arginine should be more severe in affecting the ubiquitination, if that was a ubiquitination site, than a residue that is 2 or 3 amino acids away and affecting the PK of that. So what is your thinking? Why are you hedging your bets? What do you really believe? And do you believe that you can hedge your bets?
I think I just can't make a definitive conclusion from this yet. I mean, I'm pretty sure. I'm very sure it's an XIAP binding site, and whether this site is ubiquitinated, we don't know.
How do you explain your K209R mutant data then? You've mutated the actual residue.
Yeah. Well, it must somehow have an influence on the conformational state of the protein and how other proteins bind there.
Yeah.
I don't. Yeah. I can't really give you a good answer.
Because one of the biggest problems is that lysine 209, we never saw diglycine marks, but actually we never saw it at all. So we can't really tell that it's not ubiquitinated at the moment as well. So we don't be missing. It never flew on an aspect. You tried.
But K209R cannot be ubiquitinated at that residue.
I agree. Yeah. So this clearly shows all the ubiquitination sites are important and do something. But I think at the moment we're not in the position to completely refute that 209 gets ubiquitinated because we just don't have the data. We don't. But this is tricky. All right, so please join me in thanking Valentin for a great seminar.